Okay, so let's take a trip back in time and recall some of this general biology as we explore carbohydrate metabolism. So dietary carbohydrates are burned as fuel very quickly after absorption. All three of the monosaccharides are going to be absorbed from digested food. As you know, we've got glucose, galactose, and fructose, the last two of which are quickly converted into glucose. So all oxidative carbohydrate consumption is essentially glucose catabolism. And the overall reaction is the one we see here. So we begin with glucose, we add some oxygen and a little ignition, we set fire to it, and we release a lot of energy, plus carbon dioxide and water. So the point of this is not really to release the carbon dioxide and the water, it's more to burn the sugar. Now, this equation is exactly the same for burning sugar as it would be for burning, say, paper or wood. It's a carbohydrate in a different form. It's a form of cellulose when we look at paper or wood. And we set fire to it, we give it oxygen, it releases heat very fast, we yield carbon dioxide and a little bit of water. However, most of the energy is going to be released as heat and light. Inside the human body, when we're burning glucose, we're going to cut this down stepwise and capture as much of the energy as possible and store it as ATP, and then we release a small amount as heat. So we begin with glucose, and it's broken down in three major pathways. First of all, glycolysis. You'll remember we're taking glucose, glycolysis, and splitting it into two. So we begin with one 6-carbon sugar, and then we split it into two pyruvates or two pyruvic acids, each having 3 carbon molecules. So we still have a total of 6 carbons. It's just been split into 2 parts. After the process of glycolysis, we yield a very little bit of ATP, and we're left with this pyruvic acid, which could either, if there's no oxygen present, go through the processes of anaerobic respiration that you see over here in the purple, or we could go through aerobic respiration if oxygen is present, and we'll completely oxidize pyruvic acid into carbon dioxide and water, and we'll trap lots of ATP, somewhere in the order of 36 ATP total. The anaerobic fermentation is going to yield no further ATP, but it will recycle these electron carriers, these coenzymes, so that we can continue to fuel the process of glycolysis. So glucose catabolism is a series of small steps that's each controlled by a separate enzyme. Not all of them are pictured here, but we release energy in very small, manageable amounts and transfer as much of it as possible into ATP. In order to accomplish this, we need coenzymes. So enzymes themselves actually remove the electrons. We're basically taking the 6-carbon glucose and stripping off hydrogen atoms and cutting the bonds between the carbons to release this energy. So the enzymes remove the hydrogen atoms, and then they're going to transfer the hydrogen atoms to the coenzymes. And then the coenzymes will donate them to other compounds later on in the pathway. These enzymes that are involved in glucose breakdown can't function without the coenzymes. The coenzymes we're talking about here are NAD and FAD. So as you might remember from general biology, NAD and FAD are like little cars. And the little cars come along and pick up 2 electrons and 2 protons, which we note simply as 2 H pluses, and just understand that every time a hydrogen atom travels, it travels with an electron. And basically, they're going to transfer these atoms later to other pathways. in the oxidative pathway, electrons are transferred into the electron transport chain, and so will yield lots and lots of ATP. In the pathways of fermentation, we'll see that these coenzymes are dropping off electrons in other pathways so they can recycle and come back up to the pathways of glycolysis and pick up more electrons. So it's important to understand that hydrogen atoms are always going to be removed in pairs. So 2 protons and 2 electrons at a time. If you took my general biology class, you'll remember the prom example where we have 2 hydrogen atoms, those are the boys, and 2 electrons, those are the girls, and they each go to prom together in one limousine. And the limousines are the coenzymes NAD and FAD. And so this produces a reduced coenzyme, because remember, reduced means to add electrons, which reduces the charge. And it has a higher free energy content than before the reaction. So there's lots of excitement about prom as these 2 hydrogens and electrons, or boys and girls, are loaded in the car. So the coenzymes then carry these excited electrons with their hydrogens and deliver them to the prom or other places. So FAD binds 2 protons and 2 electrons to become FADH2. And NAD will bind the same 2 electrons, but only really truthfully one of the protons. The other one becomes a free proton as a hydrogen ion. So we say it's NADH plus NH plus. So here we have 2 NADHs plus 2 H pluses. So the reduced form of both of these are FADH2 with 2 electrons and 2 hydrogens in a car, or NADH plus an H plus, which essentially in our analogy of the prom is that we have the NADH with 2 girls and 1 guy in the car and the other guy's running down the street behind it, plus the H plus. It's kind of a funny analogy, I know, but it works. So we'll first examine glycolysis. Glycolysis, the splitting of sugar. We start with glucose up here. We'll start with glucose here. And we'll end up at the bottom with 2-pyruvic acid, or pyruvate. So we'll split glycolysis into a few more steps than we did perhaps in general biology. However, they're important steps because this is where we're going to see that metabolism of other fuels fit into the pathway. So glycolysis is a series of conversions that happen to glucose after it enters the cell. We'll begin with the phase of phosphorylation up here in the red. Phosphorylation is where we see the enzyme hexokinase transferring energy or inorganic phosphates from ATP to the glucose. Both phosphorylation and priming are the energy investment phases of glycolysis. The addition of the phosphate group generates the product glucose 6-phosphate. And the purpose of having glucose 6-phosphate is twofold. First of all, it keeps the intracellular concentration of glucose quite low. Thus, we favored continued diffusion of glucose into the cell to supply the chain of glycolysis. It also prevents the sugar from leaving the cell because these phosphorylated compounds can't pass through the plasma membrane. This first step is irreversible because cells lack the enzyme to convert glucose 6-phosphate back into glucose. There are a couple of exceptions though. In cells that have to be able to release free glucose into the blood, such as the absorptive cells of the small intestine, in the proximal convoluted tubule cells of the kidney, and in liver cells. But for the most part, cells cannot go backwards on this step. Glucose 6-phosphate is a very versatile molecule. It could be converted to fat, or it could be converted to amino acids. It could be polymerized for glycogen for storage. And it could be further oxidized to extract energy, which is the pathway that we'll follow. The next step in the game is priming. Now, priming is much like if we wanted to set our log on fire or a piece of paper on fire, we would have to add a little energy, like a spark, use a lighter, a match. And so here we're going to add a little energy, and just like when we burn paper, we release much more heat than the initial energy invested because we're releasing stored energy in this carbohydrate compound. So in priming, we're going to rearrange glucose 6-phosphate into an isomer to form fructose 6-phosphate here, which is then going to be phosphorylated by the addition of some phosphate and energy from ATP to reveal glucose 1,6-diphosphate. And this provides the activation energy or the spark necessary to set this sugar on fire so we can capture as much energy. Cleavage is the next phase in the game. This is the lysis part of glycolysis. It's where we split this 6-carbon molecule, fructose 1,6-diphosphate, into 2 3-carbon molecules, phosphoglyceraldehyde, which is otherwise known as glyceraldehyde 3-phosphate. You may have seen it before as G3P. These 2 glyceraldehyde triphosphate or phosphoglyceraldehyde molecules will then continue on down through the pathway to finally reveal pyruvic acid. You'll notice in the steps from here on it says 2 times this molecule and 2 times this molecule. So we're continuing illustrating these 2 molecules just in a single train, but by adding 2 out in front. So following cleavage, we have oxidation. And remember, oxidation means to remove electrons. Reduction, adding electrons. Oxidation, removing electrons. So each PGAL molecule is then going to be oxidized by removing a pair of hydrogen ions. This is where our coenzyme NAD comes in. NAD here comes in, picks up 2 hydrogens, which always travel with electrons, and picks them up and takes them off to another destination. We'll explore that later. In addition, we're going to pick up a phosphate ion here that's floating around in the cell's pool of free phosphate ions. This one doesn't come from an ATP. Now, we'll move into dephosphorylation. This is where we're going to strip the phosphates off and add them to ATP. So the phosphate groups are taken from intermediates in glycolysis. We're not worrying about what their names are here. And they're going to be transferred onto ADP. So ADP comes in here, picks up phosphate to become ATP. So this is the first place that we're seeing capturing of the energy that we release from breaking apart these carbon-carbon bonds. And we have some energy stored in the ATP now. And we also have these high energy electrons stored in our limousines or in our cars that are picking up the hydrogens and the electron pairs. At the end of dephosphorylation, these 2 3-carbon compounds have become pyruvic acid. And pyruvic acid is now the precursor to either of 2 pathways, either anaerobic fermentation or aerobic respiration. So the net products of this whole thing are 2 ATP, 2 NADH plus the 2 free hydrogen ions, and 2 pyruvic acid. So it's very important to keep track of what the products are of each phase. So these are the products of glycolysis. We have the same energy originally in glucose contained in the ATP, some in the NADH, little bit got lost as heat, but most of the energy still remains in this pyruvic acid. And this is what we're going to see release so much ATP should it go into the pathway of aerobic fermentation. Remember that we invested 2 ATP and we received 4 ATP. So the net production from glycolysis is 2 ATPs. Go ahead and pause the recording for a moment and take time to diagram out the process of glycolysis. Each of the phases, phosphorylation, priming, cleavage, oxidation, and dephosphorylation. And remember, the important things to track here are: where do the carbons go? Where are the electron carriers, these coenzymes? And have they picked up or dropped off electrons? How many ATP? So we're tracking carbons in the pyruvic acid, stored energy in the coenzymes, NAD and FAD, and then stored energy in the form of ATP. So go ahead, diagram that. It'll be part of the diagramming assignment for this chapter. So now that we have this intermediate of 2 pyruvic acids, high energy containing molecules, there's a couple of possible fates, and they depend upon the availability of oxygen. So in an exercising muscle, there's an increasing demand for ATP, and that demand might exceed the oxygen supply that's available, especially at the beginning of exercise, because the lungs haven't had time to catch up. So the only ATP cells can make at this point is produced by glycolysis. And as you've already discovered, that's only 2 ATP, so it's not very efficient. And glycolysis can't continue indefinitely because we're going to run out of NAD cars to pick up electrons. And in order for glycolysis to continue, we need to continue to pick up electrons from the original 6-carbon molecule so that we can continue to produce pyruvic acid. So the process of anaerobic fermentation is how we return NAD back to glycolysis so that it can continue to pick up electrons and hydrogens, thus removing them from the original glucose and allowing the continued production of pyruvic acid, and thus the production of ATP. So NADH comes from glycolysis and moves to actually add a pair of electrons and hydrogens to pyruvic acid, the end product of glycolysis. As you add those electrons and hydrogens, it reduces the pyruvic acid into lactic acid. And you've probably heard of lactic acid. It's the stuff that makes our muscles sore during exercise. But as we drop off the electrons and the hydrogens to pyruvic acid and convert it to lactic acid, the electron cars, the limousines, are emptied of their electrons and hydrogens so that they can return to glycolysis and pick up more. So the whole purpose of anaerobic fermentation is to recycle or regenerate NAD so that glycolysis can continue. However, you understand already that we can't continue to build up lactic acid indefinitely. To start with, the lactic acid can leave the cells that generate it. It'll enter the bloodstream and it'll get transported to the liver. And at the liver, when oxygen becomes available, that'll be oxidized back to pyruvic acid and it can be returned to the pathway of aerobic respiration. The liver can also convert lactic acid back to G6P. That's the phosphorylated version that can't leave the cell. And then it can polymerize it to form glycogen, which is our storage form of glucose. It could also remove the phosphate group and release the free glucose into the blood to add to blood sugar. However, as you know, there are drawbacks to anaerobic fermentation. It can't go on together because of this byproduct, lactic acid. Lactic acid's toxic to the tissues and contributes to the muscle fatigue. It's also wasteful Because remember, most of our energy is still tied up in these carbon-carbon bonds that we see in pyruvic acid. They're still there in lactate. We need to reclaim that energy and put it into the process of aerobic respiration. Now, skeletal muscle, it's fairly tolerant of anaerobic fermentation, but cardiac muscle is much less tolerant. And it's good to note that the brain employs no anaerobic fermentation because we simply don't want lactic acid there. Another thing of note is that during childbirth, when the infant's blood supply is cut off, almost every single organ of the body switches to anaerobic fermentation. And this allows sparing of oxygen for the brain's metabolism so that it can continue to function. So none of the organs are competing with the brain for this limited supply of oxygen. So this brings us to the end of this section of carbohydrate metabolism. So take a moment to diagram the process of anaerobic fermentation and consider how NAD is recycled so that we can continue the process of glycolysis and the production of pyruvic acid. Include the conversion of pyruvic acid to lactic acid. So pretty much you're going to put into your own figure the contents of this purple box in the figure to the right here. And we'll see you in the next section of carbohydrate metabolism.